Okay, so for today, it's just an overview of carbohydrate before we jump into learn about the carbohydrate metabolism. So as for protein, last week we learned about the structure, the basic unit of the protein. So today we are going to learn about the one of the largest biomolecules, which is the carbohydrate. Okay, so this is a part one for the carbohydrate. The second part will be the carbohydrate metabolism. Okay. So by end of this lecture today, you should be able to define the term of monosaccharide, disaccharide, oligosaccharide, and polysaccharide, and explain the structure of carbohydrate of physiological significance, and also describe the main role of carbohydrate in providing and storing the energy, right? Okay. So what is polysaccharide that made up our carbohydrate? So it's a polymer, which a large molecule that made up by stringing together like beads on a string with smaller unit called the monomer. So poly means many, and polysaccharide are made by a chain of monosaccharide monomer. Okay, and monosaccharide is the sugar, which is the monomer for polysaccharide. So the carbohydrate are the most abundant bioorganic molecules in nature. So it consists of the carbon, hydrogen, oxygen that bond together. So this is the empirical formula for the carbohydrate, C and H2O, and the number of the atom. So it comprises of carbon and water. So the main role or function of carbohydrate is to provide energy in the diet of most organisms. So human will break down the carbohydrate using the during the process of metabolism to release the energy. So this is an example of the reaction when glucose is breaking down to form the carbon dioxide and water and also energy. Okay, so we will learn in detail how the breakdown of carbohydrate in carbohydrate metabolism lecture. So human obtain carbohydrate by eating foods that contain them, for example, potatoes, rice, breads, and etc. And carbohydrate are manufactured by plant during the process of photosynthesis. So the metabolic pathway of the photosynthesis require energy from the sunlight. That is where the glucose is made up. Okay. Okay. What are the main function of carbohydrate? So it acts as a storage form of energy in the body, and it serves as a cell membrane component and as a structural components of many organisms. So it built the cell wall of bacteria, exoskeleton of insects, and also made up the fibrous cellulose of plants, right? So carbohydrate have distinct classes, four main size of classes based on its number of sugar unit. So monosaccharide is the simple sugar, consists of single polyhydroxy aldehyde or ketone, and most abundant is the glucose or in a configuration of D, the D glucose or dextrose. And disaccharides consists of two monosaccharide units joined by glycosidic bond. And the common one is the sucrose, which made up by the glucose and the fructose. And oligosaccharide is a short change of monosaccharides unit. Usually it has the 3 to 10 residues joined by the glycosidic bond. And polysaccharide is a long chain sugar polymer, comprises more than 20 to 100 or 1,000 monosaccharide units. So usually cellulose, starch, and glycogen is a type of polysaccharide. And also we have the glycoconjugate, a more complex structure, a more complex carbohydrate usually attached with protein or lipids. That is the glycoconjugates. Okay. So before we start to learn about the different class of carbohydrate, so let's learn the naming or the drawing of different types of carbohydrate units. Okay, so usually Fisher projection is used to distinguish the different stereoisomer in a open chain or linear form. So you have to place. So if you look at this Fisher form, Fisher projection, so the carbonyl group is placed at the top here. Okay, and then at the bottom you will have the non-chiral CH2OH carbon in here. So the backbone is made up by the chiral carbon. So for this glucose, it has 6 carbon. 1, 2, 3, 4, 5, 6. Okay, so the carbonyl is placed at the top and then non-chiral CH2OH is placed at the bottom. So this is the Fisher, what we call it, a Fisher projection drawing. Okay, so it gives us information of the types of the carbonyl group, whether it is aldehyde or ketone, that we will learn after this, and also the number of the carbon that made up the backbone of the one monosaccharide unit. Okay, so in general, a molecule with a chiral center can have 2N isomer. Means, let's say this is 6 carbon. So the potential isomer that can be formed by glucose is 2 to the power of 6. For example, this cholesterol have 8. 1, 2, 1, 2, 3, 4, 5, 6, 7, 8 chiral carbon. So the potential stereoisomer that it can have is to the power of 8, 256 different isomer. But nature only can make only one. But the possible stereoisomer, we learned about the stereoisomer previously, right? So the potential it has is to the power of 8. Depends on the chiral carbon that made up the backbone. Okay. So in Fisher projection also, it gives us the information about the configuration of the units, whether it is in the D or L configuration. Okay, so it is based on the uh, chiral carbon, which is far or further from the carbonyl group. So let's say if you look at this glucose here, the glucose, so the carbonyl group is here. So where is the furthest chiral carbon? So this is 1 chiral carbon, 1, 1, and this is the furthest chiral carbon from the carbonyl group. So based on this chiral carbon, you should decide whether it is in the D or L configuration. If the hydroxy group is at the right, so it is a D isomer. So this is a structure of glucose, so it should be the D glucose. So if the hydroxy group is in the left, so it is L isomer, L glucose. So this is the enantiomer of the glucose in D and L configuration. Okay. So this is very basic. I think all of you should already recognize how to differentiate between the D and L configuration. So we learned previously in the protein. So this is in the carbohydrate. Okay. So this is the Fisher projection, and you should know how to name. So if I give this structure and I want you to name it, name all the stereoisomer, then you should be able to do it based on the carbonyl, whether it is aldehyde or ketone, and also whether it is in a D or L configuration. Okay. Okay, let's see another example here. So the simplest aldose is glyceraldehyde. So it contains only one chiral center. So here the structure of the glyceraldehyde. So it have only one chiral center here. So what is the chiral carbon? The chiral center, carbon that attached to 4 different group. Okay, carbonyl, HOH, the hydroxyl, and also the CH2OH, the non-chiral carbon. So this is only one chiral carbon. So what is the potential of the isomer that glyceraldehyde can have? So 2 the power of 1, only one chiral center, so it only have 2 isomer. or enantiomer. Okay. So L and D configuration also can get information from the Fisher projection, as I mentioned. So this is the OH placed at the right. So it is D-glyceridehydrogenide. And it mirror image, the isomer is in L-glyceridehydrogenide because our hydroxy group is in the left. Okay. So that is how you read the Fisher projection and name the molecule. Okay. So let's say, for example, you go back and try to do a drawing of Fisher projection for D and L for lactic acid and alanine, and we will discuss about it in the next lecture. So I will take a few time and we can discuss about this one. So go back and try, okay, to name this based on the Fisher projection. Okay. And I provide this as well, so you can go and have a look. Okay, let's see the first class of the carbohydrate, which is the simplest sugar, the monosaccharides. So monosaccharides are colorless crystalline solid. It is freely soluble in water, insoluble in non-polar solvent, and it is a basic unit of the carbohydrates. And most of them have a sweet taste. And so you have to know the monosaccharide can have the aldehyde or ketone functional group. So from this aldehyde and ketone, we will further classify the monosaccharide. Okay, so what is aldehyde and ketone? So for the classification of the monosaccharide, it can be based on their carbonyl group here. So you should identify whether the carbonyl group is the aldehyde. So the carbon bonded, double bonded with O, bond with the R, and also the H, the hydrogen. So this is the aldehyde. But for ketone, carbon double bonded O with both R. So this is the ketone. Okay, you should identify in the structure whether it is the carbonyl is in it has the functional aldehyde or functional ketone. Okay, so usually aldehyde, the carbonyl group is at the end of the carbon chain here, and the ketone usually at any other position. Okay. Okay. So if the carbonyl group is have the aldehyde functional group, so it is known as aldose sugar. So example of the aldose sugar is glyceridehydrogenide and glucose. And if the carbonyl group have the ketone functional group, it is in the class of ketose sugar. So for example, the dihydroxyacetone and also fructose. That is the example of ketose sugar. So you can recognize based on the prefix aldo and also for the ketone, the prefix keto. So let's see the example. Okay. So monosaccharide also can be classified according to its number of the carbon. Okay. So the simplest monosaccharide are the 3 carbon, the trioses. Okay. So the trioses have 3 carbon in the backbone. Example, the glyceridehydrogenide, which is an aldose, and dihydroxyacetone, which is a ketose. So it have a ketone functional group, and glyceridehydrogenide have the aldehyde functional group. Okay, another example is the glucose. So glucose have the aldehyde functional group, C double bonded bond with R and also the hydrogen, and it have 6 carbon in the backbone. So it is known as aldose hexoses sugar. Okay, and for ribulose, it is in the ketopentose sugar because it has the ketone functional group and also 5 carbon, number of carbon in the backbone. Okay, so let's see for example here. So this is the aldehyde functional group. Okay, 1, 2, 3, 4, 5, 6 carbon in the backbone. So it is glucose with aldehyde functional group and 6 carbon. So it belongs to aldohexoses. Okay, and this is for ribulose. It has the functional group of ketone and consists of 5 carbon in the backbone. So ribulose belongs to ketopentose sugar. That's it. Very straightforward. Okay. Okay, so I also give an example here. Please identify and classify each of these monosaccharide, okay, according to its class. Then we will discuss about it next week, inshallah. Okay. So this is an example of the D-aldoses. So D-glyceridehyde have one only chiral carbon. So the potential isomer it have only two, the D-glyceridehyde and L-glyceridehyde. And for example, the erythrose here and D-triose, ribose, arabinose, xylose, and lysose also a type of D-aldoses. So it have the functional group of aldehyde here at the top, okay, based on the Fischer projection. Okay, so also this is a family of aldoses. So it have a potential 16 isomer to the power of 4. How they got this? So based on the number of carbon, a chiral carbon, 1, 2, 3, 4. Okay, to the power of 4, 16. 1, 2, 3, 4, 5, 6, 7, 8. Because this is D-aldose, another 8 come from the L configuration. So 8 plus 8 is 16. So that's how you got the different isomers. Okay. So I guess this is a very straightforward, and you should already have the basic, okay, in the chemistry of carbohydrate. Okay, so this is an example of D-ketosis. Okay. Okay, so let's do this very quick. So how we can classify each monosaccharide? So can everyone answer what type of or what class this monosaccharide belongs to? Okay, so it has a functional group of aldehyde or ketone.
Ketone.
Okay, so and then it belongs to which sugar? Which
Ketones.
sugar class?
Ketose.
1, 2, 3, 4, 5, 6. Okay, ketose and 6 carbon, and then it becomes
Ketohexose.
okay. Yes, ketohexose. Very easy. I know this is very easy for you. Okay, number B. Number 2 here. So which functional group?
Aldehyde.
Aldehyde.
So aldehyde with C carbon, and then it become
Aldehexose.
Aldehexose.
aldohexose. Right, right. Okay. And this one?
Aldehyde.
Aldehyde.
So where is the functional group? Here?
Top.
Here, right? So it have 1, 2, 3, 4. 4 carbon, aldehyde. So aldo, tetrose. And how about this one? Where is the functional group?
Ketose.
So this is the functional group of ketose, right? So it have 1, 2, 3, 4, 5 carbon. So it is ketopentose. So that's how you classify each monosaccharide based on the functional group of the carbonyl, whether it is aldehyde or ketone functional group, and also the number of carbon. Okay. Okay, so you should know only 1% of monosaccharide exists in its open chain. Open chain is like this one, open chain. Most monosaccharide will exist in a cyclic form. So molecule which have the alcohol group and also the carbonyl group can undergo intramolecular reaction, what we call it intramolecular reaction, to form a cyclic. So it will transform from the open chain and then to become a cyclic ring, okay, where the alcohol group in the molecule can add into the carbonyl group, aldehyde or ketone in the same molecule to form a stable ring, which we call it hemiacetal or hemiketal. Okay, what is hemiacetal and hemiketal? So aldose sugar which have aldehyde functional group can cyclize to form a cyclic hemiacetal. So from this open chain, so it will transform to become a ring of hemiacetal. So this is hemiacetal. You can see the bonding and also the OH hydroxy group attached to the carbon. Okay. For ketone sugar, it can cyclize to become the cyclic hemiketal. So so this is aldose. So maybe you can remember aldose A here, then it become the hemiacetal. Then ketone K here become the hemiketal. Okay. So the hemiketal ring bonded to OH and also other functional group. So let's see how glucose transform from a linear open chain to a ring structure. So this is a D-glucose. D because the hydroxy placed at the right. Okay, glucose because it have six carbon atom. 1, 2, 3, 4, 5, 6. Okay, so this is the structure of glucose in Fischer projection. Okay, so what happened here? It will transform like in a clockwise. So this C will be on the right side facing downward here, and it will um, turning a clockwise. Okay, and then become a ring. So there is a new carbon formation here, carbon bonding, and this carbon formation we call it as a anomeric carbon. So because this is no bonding with other structure, right? But in ring formation, hemiacetal formation, so it will form another bond here for the carbon. And the new carbon that form bond, we call it as an anomeric carbon. Okay, so that's how the ring of the glucose is made up. Okay, so let's look at the ring of the glucose here. Why the name for Fischer projection is D-glucose, but in Howard projection, so in ring structure, we call it in a Howard projection. So beta D glucopyranose. Beta because if you look at the carbon anomer here, that carbon forming a new bond, so the hydroxy group and the CH2OH here at the same side, facing upward. So when it facing outward and have the same side, on the same side, it become a beta. So let's say the hydroxy group is here. So it will become the alpha. So the name of the cyclic glucose is beta D glucopyranose. And pyranose because it have a 6-member ring. If it has 5-member ring, and it will become the pyranose. We will learn about the ring later, but just to show you how from the linear D-glucose, it will transform to form a cyclic glucose with a name of beta D-glucopyranose. So this is a projection of Howard. Okay. So the hemiacetal and hemiketal carbon that form the ring is called the anomeric carbon. So usually for the aldose, it is at the carbon number 1. And for ketose, it is at the carbon number 2. So this is the aldose. So the anomeric carbon is at the carbon number 1. Okay. And for the ketose, so this is for aldose and this is for the ketose. The anomeric carbon is at number 2 because the carboxy group in aldehyde is at the top, right? Okay. So when it form the ring, the anomeric carbon form a new bonding. So this, the C number 1 is this anomeric carbon for the aldose. And for the ketose, the anomeric carbon is at carbon number 2, right? So you just have to differentiate that one. Okay. So the alpha anomer has the OH pointing down in the ring form. So this is the alpha. And the beta anomer has the OH pointing up. So OH pointing up is the beta anomer. So the ring formation has a dynamic equilibrium reaction, so it can form in an open chain and then transform into the cyclic ring and then form again its open chain. So it can switch back and forth between the two ring form. Okay. Okay, the 5-member ring hemiacetal with oxygen at the top here, it is known as the furanose because it have a 5-membered ring. Okay. An example is the fructose, ribose, and deoxyribose. And for the pyranose, it has 6-member ring, which is formed in a hexagon. So the oxygen is the upper right, and hemiacetal carbon or the anomeric carbon is placed at the right. For example, the glucose, galactose, and also the mannose. Okay, let's see, for example, the glucose anomer. So it is in a pyranose cyclic form of glucose because it has 6 carbon. 1, 2, 3, 4, 5, 6. Okay, and OH is pointing down. So alpha hydroxy group and OH pointing up is the beta hydroxy group. So this form are anomeric of each other, and carbon 1 is the anomeric carbon in the glucose anomer. Okay. So the OH at the right in the Howard projection, if it is placed up. Okay, so if it is in the left, it will place down. Okay. So place up is the beta, down is the alpha. Okay. So because of the CH2OH, the non-chiral carbon, the last carbon is placed up. So it is in a form of D configuration. So if it is placed down, it is in the form of L. Okay, how about the fructose anomer? So it can form a 6-member pyranose ring by reaction of the C2 keto group, C2, with the hydroxyl on the C6 of the backbone, and also it can form a 5-member pyranose ring. Okay, so it can form both furanose and pyranose. Depends on the interaction of the C2 with the C5 or C2 with the C6. Okay. If C2 carbonyl react with the 6, okay, then it will form the pyranose ring. Okay, so if the C2 react with the C5, it will form the furanose ring. Okay. So monosaccharide have different types of reaction that can occur. For example, the oxidation. So aldehyde and ketone that have the hydroxyl group on the carbon next to the carbonyl group will react with a basic solution of the copper ion, which we call it a Benedict reagent, to form a red-orange precipitate of copper oxide. So with this reduction by the oxidation, so we call it the sugar. We call it the sugar is the reducing sugar because it have the capability of reducing the copper ion to the copper oxide. So why it is important? Okay, let's see the effect of acid on carbohydrate. Let's say it is disaccharide or polysaccharide. When there is a stimulus by the heat or the acid, it will be break down into its monosaccharide, the pentose or hexose, releasing a H2O, and then the ring formation will be in a precipitate form, in a colored form. So why the color is important? Because we want to detect the presence of the carbohydrate in a sample. For example, if we want to detect the glucose using a Benedict reagent, if the glucose is present, then there is a yellow then red color precipitation because of the oxidation that occur in the reaction. Okay. So Benedict test to distinguish between the reducing and non-reducing sugar. So reducing sugar will have the free aldehyde or ketone group that can be oxidized by heating or with Benedict reagent. Okay, so it lead with the precipitation of the yellow and red color because the copper ion is reduced to oxidized to the copper oxide. Okay, so it will give the red color. So this is more to a technical experimentation. Okay, so let's see the structure. Here, the aldehyde sugar are readily oxidized. So this is the with the aldehyde monosaccharide with the aldehyde group, and then it will reduce the copper ion to the carbonyl group. So this is the aldehyde sugar with the aldehyde group, and then it will reduce the carbonyl group with the aldehyde group. So this is the aldehyde monosaccharide with the aldehyde group, and then it will reduce the carbonyl group with the aldehyde group. copper ion in the Benedict reagent to copper oxide. Okay. So if let's say the monosaccharide unit have the ability to reduce copper ion to copper oxide, it means that it is belong to a reducing sugar class. Okay, so this is another one example how to classing the monosaccharide. So let's say I ask you what are the types of the reducing sugar, then you should be able to identify which one is the reducing sugar and also non-reducing sugar. Okay. So basically, in diagnostic technique, it provide a good test for to detect the presence of glucose in the urine. Okay. Okay. How about the formation of the phosphate ester? So it can form at the six carbon of aldohexose and also aldoketosis. The phosphate ester of monosaccharide are found in the sugar phosphate backbone in DNA and RNA, also in ATP and as intermediate in the metabolism of carbohydrate in the body. So this is the monomer units, and it have the phosphate sugar phosphate bone, sugar phosphate group that attach to the backbone. So this is how the reaction of the monosaccharide. It can have the reaction with the phosphate group, where phosphate is added into its backbone. And the last one is the formation of the glycoside. So the hemiacetal and hemiketal form of monosaccharide can react with alcohol to form the acetal or ketal structure, which we call it as a glycoside. So a new carbon form is by the binding of glycosidic bond. And once glycoside is formed, a ring can no longer open to the open chain. So when it is in cyclic, so it has no exposed carbonyl to be oxidized. So it is in a form of non-reducing sugar. So glycoside not able to reduce the copper ion to copper oxide. Okay. So let's see here the glucose, let's say react with the alcohol. Okay, so it will form a methyl alpha D glycopyranoside and methyl beta D glycopyranoside. So this is the interaction between the alcohol. So you should know that when glycoside is formed, it is no longer belongs to a reducing sugar class. Okay, because it's in a ring formation. Okay, this is an example of important types of monosaccharides: beta D deoxyribose, beta D ribose, and beta D galactose. And I give an example here so that you can study about its structure, how it got its names by the ribose D configuration, and how it gets the beta here. Okay, beta anomer. So maybe you can go back and then learn about the naming. Okay, by the illustration that I provide here. Okay, and also this is an example, beta D glucose and beta D fructose. Okay, so now let's move on to the second type of carbohydrate, which is the disaccharide. So it consists of two monomer joined together by glycosidic bond. Okay, so it form when a hydroxyl group on one of the sugar react with the anomeric carbon on the second sugar, and then the linkage form is the glycosidic bond. Okay, and most common are maltose, lactose, and also the sucrose. So in maltose, it is known also a malt sugar, and it formed during the hydrolysis of starch to glucose during digestion. Okay, and it belongs to hemiacetal group, and hemiacetal have the aldehyde group. Aldehyde have the carbonyl uh, group that can later be oxidized. So all aldehyde is hemiacetal, and all hemiacetal are reducing sugar. Okay. Okay, so you have to remember that. Okay. And two D-glucose joined by glycosidic linkages between the C1 of anomeric carbon of one glucose and then react with the C4 of the second glucose. So this is the first glucose, second glucose, the first C carbon, the anomeric will react with the C4. 1, 2, 3, 4 C carbon on the other glucose. And it will form the glycosidic linkage. So this is the disaccharide of the maltose. So if I ask you how maltose is made up, it is made up by the interaction bonding between two glucose by glycosidic linkages. Okay. Okay. So the configuration of anomeric carbon in glycosidic bond between two glucose is in the alpha. So lactose, also known as a milk sugar, it consists of the galactose and glucose joined together by glycosidic linkages. So the C1 of the galactose interact with the C4 of the glucose. Okay, so that's how it is formed. So the configuration of anomeric carbon in glycosidic is in the beta. It follows the anomeric carbon of beta D galactose. Okay, so the configuration of the disaccharide here is in B configuration. Okay, depends on the anomeric carbon of the galactose. Okay, so it also belongs to the reducing sugar class since it has one free carbonyl group on glucose residue that can be oxidized. So you just remember all the aldehyde in hemiacetal is reducing sugar. How about sucrose? Sucrose is known as the table sugar. So it's joined by the glucose and also the fructose. Okay, the C1 on the glucose react with the C2 on the fructose by a glycosidic bond. Okay. Okay. So the configuration of the glucose in alpha, the glycosidic of the anomeric hydroxyl in fructose is the beta. So the linkages is alpha 1, beta 2. Okay, so because both are anomeric carbon. Okay, so this is anomeric carbon of the fructose and this is anomeric carbon of the glucose. So alpha glucose at carbon 1 react with the beta glucose at carbon 2. So the naming will be the alpha 1 beta 2 based on the configuration. Okay. So it is also belong to reducing sugar since it has one free carbonyl group on the glucose. Okay. Okay. So the sucrose is the sweetest of the three common disaccharide. Okay. So next one is the oligosaccharide. So it consists of 3 to 10 monosaccharide units linked by the glycosidic linkages. Okay. For example, the raffinose, beta D galactose interact with the glucose and also the fructose. So all these three monosaccharide units form the raffinose, and this is a type of oligosaccharide. Okay. So the most important other than the monosaccharide is the polysaccharide. The complex polymer of 10 or more could be the 100 or 1,000 monosaccharide units. So it can be divided into the homoglycan, which have the homopolysaccharide composed of only one type of monosaccharide. And also the heteroglycans, heteropolysaccharide made of more than one type of monosaccharide. Okay, it can have the linear or branch chain, and it also belongs to the non-sugar, non-reducing sugar because no anomeric carbon are connected through glycosidic linkage. No free anomeric carbon to be oxidized. Okay, and for example, the starch, so the polymer of the glucose unit. Okay, and it is belongs to the homoglycan type. There are two forms, amylose and amylopectin. So amylose is a long linear polymer of glucose unit linked by alpha 1,4 glycosidic bond. So alpha from one glucose at C1 interact with the C4. So it structure naming will be alpha 1,4 glycosidic linkage of the glucose in starch. That is for amylose. And for amylopectin, it is long branch polymer and the backbone of glucose linked with alpha 1,4 glycosidic bond, and the branch point, which is every 24 to 30 glucose unit, will be linked at 1,6 glycosidic bond. So this is a linear form. like the amylose, but when it have a branch at the alpha 1,6, so alpha 1 C1 interact with the C6 of another glucose, and it will become the alpha 1,6 branch point. Okay, that is amylopectin, which is the branch polymer, more complex compared to the amylose. Okay. Right, so this is just illustration. Amylose is single unit of glucose bonding together by the glycosidic bond. But for amylopectin, it have branching at the alpha 1,6. Next is glycogen, also known as animal starch. So it is as a storage form of glucose in animal cell. It is branch polymer of glucose, which is also belong to homoglycans class. It is similar to amylopectin. So the backbone of glucose link at the alpha 1,4, same like amylopectin. The branch also link at alpha 1,6, but the difference is that it is more highly branched because the branching occur every 8 to 13 glucose unit. For amylopectin, 24 to 30, and then there is one branch. So it is more highly branch of the glycogen. So it occur every 8 to 13 glucose unit. Okay, so that is the difference between the starch and also the glycogen. Okay, how about the cellulose? It is a structural polysaccharide. It made up the plant cell wall, linear polymer of long, unbranched homoglycan chain of D-glucose, linked by the beta 1,4 glycosidic bond. Okay, so the beta 1,4 glycosidic bond is very stable and difficult to hydrolyze, and also extensive of the hydrogen bonding within and between the cellulose chain. Okay, so it makes a bundle of fibril, so it will become the rigid structure. So that's why it's made up the plant cell wall. It is known as the structural polysaccharide. Okay. Okay, that is cellulose. So different between the starch, glycogen, cellulose is you can see at the glycosidic linkage, the place of the glycosidic linkage, and also the branching. Okay. So you need to identify between those three. Okay. Okay, so the last one, the type of the heteroglycans, which it makes the glycoconjugate, which is the derivative attached to protein, lipids, or peptide. So monosaccharide can attach to other proteins, lipid, or peptide, so that it is known as heteroglycan. Okay, not only made up by only monosaccharide monomer. Okay, for example, the proteoglycan, it is a complex polysaccharide called glycosaminoglycan. It is and core proteins. It is found in extracellular matrix of connective tissue, and it is unbranched heteroglycans of amino sugar, galactosamine or glucosamine, and alduronic acid. So this is how it made up. For example, the hyaluronic acid found in cartilage and synovial fluid, and also example of the proteoglycan cartilage. Okay, so this is example of a complex polysaccharide. And what about the peptidoglycan? So it is found in cell wall of bacteria. So it composed of N-acetylglucosamine and acetylmuramic acid joined by beta 1,4 linkages. And for glycoprotein, it is oligosaccharide attached. Carbohydrate change are from 1 to 30 residue in length. Example, the enzyme. Hormones are glycoprotein, structure protein, transport protein, and it is also found in eukaryotic cell. Okay, so can be attached to protein with a 1 or 2 configuration, or link carbohydrate bonded to a hydroxy group of serine or threonine. So it make a link with the protein. N-link carbohydrate usually and acetylglucosamine link to the asparagine. Okay, so I give an example, 3 heteroglycans. So just for you to know that polysaccharide are not made up only with a same type of monomer. So it can attach to long linear or branch chain, can attach to other biomolecule as well. Okay, for example, the peptide. This one is protein, and also the lipids. Okay. Oops. Okay. So this is example of heteroglycan. Will be the last slides for today.